Bismillahirrahmanirrahim. So let's understand the synthesis of red blood cells, also known as erythropoiesis. So the production of red blood cells starts in the early weeks of gestation, and the site of red blood cells production is yolk sac. During the middle trimester, the primary organ for the synthesis of red blood cells is liver, whereas spleen and lymph nodes also produce significant amount of red blood cells. In the last month of gestation and after the birth, red blood cells are mostly produced by the bone marrow of almost all the bones. And after the 5 years of age, the bone marrow of vertebra, sternum and ribs are the most common sites for the production of red blood cells. So in the last month of gestation and after the birth, red blood cells are exclusively produced in the bone marrow. So red blood cells originate from stem cell colonies known as pluripotent hematopoietic stem cells. These pluripotent hematopoietic stem cells undergo cell division, forming more of their own kind, whereas some of these stem cells differentiate into a specific colony of cells known as colony-forming unit spleen cells or CFUS. These colony-forming unit spleen cells produce all the blood components, including red blood cells and white blood cells. For the formation of red blood cells, these colony-forming unit spleen cells further differentiate into colony-forming unit erythrocytes. Erythrocytes mean red blood cells. So these colony-forming unit erythrocytes are committed to produce red blood cells, and therefore they are also known as committed cells. These colony-forming unit erythrocytes form red blood cells and no other cell. And this formation of red blood cells is regulated by a specific hormone known as erythropoietin. So erythropoietin upregulates the synthesis of red blood cells by acting upon colony-forming unit erythrocytes. So coming on towards the regulation of red blood cell synthesis. So the main regulator for red blood cell synthesis is tissue oxygenation. So low blood oxygenation, anemia due to decrease in the quantity of red blood cells, low concentration of hemoglobin, and excessive blood loss can all lead to decreased oxygenation of the tissues causing hypoxia. The hypoxia of renal tissues is by far the most important driving factor for the production of red blood cells. So hypoxia of the renal tissues results in the production of a specific transcription factor known as hypoxia inducible factor 1 or HIF1. The hypoxia inducible factor binds to hypoxia response element gene, which is present in proximity to erythropoietin gene, and it upregulates the production of erythropoietin by the renal tissues. Erythropoietin is a specific glycoprotein, and almost 90% of erythropoietin is produced by kidneys and 10% is produced by liver. In response to hypoxia, the erythropoietin synthesis begins within minutes to hours, and it reaches maximum production within 24 hours. The erythropoietin acts on colony-forming unit erythroblasts and upregulates the differentiation of colony-forming unit erythroblasts into proerythroblasts, which further mature into red blood cells. The erythropoietin not only just upregulates the differentiation of colony-forming unit erythroblasts into proerythroblasts, but it also speeds up the process of red blood cells maturation. So erythropoietin acts on colony-forming unit erythroblasts and upregulate the differentiation, increase the formation of proerythroblasts. The colony-forming unit erythroblasts require vitamin B12 and folic acid for the nuclear maturation and DNA formation. The deficiency in vitamin B12 or folic acid can result in megaloblastic anemia. So the colony-forming unit erythroblasts differentiate into proerythroblasts, and the proerythroblasts excessively uptake iron for the production of hemoglobin. The deficiency of iron results in hypochromic microcytic anemia. So proerythroblasts further differentiate into basophil erythroblasts. They are known as basophil erythroblasts because they stain with the basic dye. Moreover, the basophil erythroblasts contain nucleus and they contain minimal hemoglobin. The next step is the conversion of basophil erythroblasts into polychromatophil erythroblasts and orthochromic erythroblasts. With each step, the size of the nucleus is decreasing and hemoglobin concentration is increasing. The orthochromic erythroblasts convert into immature red blood cells known as reticulocytes. The reticulocytes contain nuclear remnants and remnants of certain other organelles such as mitochondria and Golgi bodies, whereas the endoplasmic reticulum is completely absorbed. The reticulocytes are released into the blood by a process known as diapedesis. Diapedesis refers to squeezing of reticulocytes through the capillary fenestrations into the bloodstream. These reticulocytes remain in the blood for 1 to 2 days, during which the nuclear remnants are completely absorbed. Hemoglobin synthesis continues during the reticulocyte stage, and all the cellular organelles completely absorb, forming the mature red blood cells, which are round, biconcave, and completely filled with the hemoglobin. The mature red blood cells are round, they are biconcave, and the average diameter of a normal red blood cell is 7.8 micrometer, and the average thickness is 2.5 micrometers. The mature red blood cells does not contain nucleus or any other organelle, and it is completely packed with the hemoglobin. The lifespan of mature red blood cells is almost 120 days, after which they are sequestered in the spleen and they are destroyed, and the iron present in the hemoglobin is returned to the bone marrow, and newly formed red blood cells take their place in the blood. So for a quick recap, first of all there is hypoxia, and most significantly hypoxia of renal tissues results in the release of erythropoietin, which acts on colony-forming unit erythroblasts and upregulates their differentiation into pro-erythroblasts, forming red blood cells. Vitamin B12 and folic acid are needed by colony-forming unit erythroblasts, and the uptake of iron begins at pro-erythroblast stage. The reticulocytes are released into the blood, and they remain in the blood for almost 1 to 2 days before they convert into mature red blood cells. The hemoglobin synthesis continues at the reticulocyte stage, and in the mature red blood cells, there is no nucleus and no cellular organelle. So this concludes our discussion about the synthesis of red blood cells. If you have any questions, do let us know in the comment section. Thank you.